Hello, everybody. Welcome to IVF Worldwide webinar today. We have today, and we are honored to host Professor Christian Egarter, who was graduated at the University of Innsbruck in 1981 and completed his gynecological residency at the Medical University in Vienna in 1988. Since 2011, he is the head of the Department of Gynecological Endocrinology and Reproductive Medicine at the Medical University of Vienna. Professor Egarter published more than 270 papers in national and international journals. His research efforts have earned him several national and international grants. The topic for his presentation today is progesterone, mode of action. Please, Professor Egarter.
Thank you very much, Seth. Ladies and gentlemen, progesterone is the topic of today. Progesterone is a steroid hormone, and the parent compound is cholesterol. Cholesterol is quite an ancient substance. It could only be built, this ring structure, when oxygen tension was around 3 to 4% on Earth. And that is why cholesterol, as well as the resulting steroid hormones, are practically present in every animal species. So progesterone has a great impact also in the human. And as you know, progesterone needs a receptor to work within a cell. And we have actually two main types, the progesterone receptor A as well as B. Both display a highly conserved DNA binding domain and a less well conserved ligand binding domain. And the only difference between the two receptors is an additional activation function 3 in the progesterone receptor B. Actually, there is also another progesterone isoform, the receptor C, that is less well described. It doesn't display a DNA binding domain, and it's only present during pregnancy. So obviously, this subtype C has more a regulatory and modification activity. Actually, you have to imagine a progesterone receptor as a three-dimensional structure. And upon binding to an agonist, there is a conformational change. Especially this helix 12 structure is moving to a different position. And that in turn leads to free areas on the surface that may further interact with so-called cofactors. So what happens when a progesterone molecule comes into the cell? It binds to the progesterone receptor upon dissociation of so-called heat shock protein 19, which are chaperones, and then there is dimerization and direct transfer into the nucleus to the DNA, the so-called progesterone response element. The next step is the activation of different cofactors, either co-activators or co-repressors, and the final step is the activation of the basal transcription apparatus, mainly the RNA polymerase II, and that leads to the activation of transcription of different genes. So what makes the situation more complex is that depending on the tissue you are, we have different concentrations of co-activators or co-repressors. We know to about 100 different cofactors, and for instance, steroid receptor co-activator 1 is abundantly present within the endometrium, whereas subtype 3 is higher concentrated in breast tissue. So depending on the concentration of the cofactors, there might be some modifications of transcription in various tissues. And to see that the progesterone impact is really tremendous, it became clear when you watch the progesterone response elements throughout the many different chromosomes. As you can see here, there is a high number of progesterone docking stations at different DNA residues. So a very recent and highly interesting animal model demonstrates the huge impact of progesterone on different pathways. In this slightly cruel animal model, the frog Xenopus levis was used, and when you cut off one extremity, for instance, the hind limb of this frog, and put a reactor to a wearable reactor with progesterone dissolved in hydrogel for 24 hours, in the months to come, there is a complete restoration of this extremity, scar-free restoration involving not only nerves to grow, but also bones as well as vessels. And that is due to the many different growth stimuli that progesterone initiates in the various pathways. So that is, in my eyes, highly interesting. But there is not only the classical pathway, the interaction of progesterone with the progesterone receptor and then the transfer to the DNA. This progesterone receptor complex may also interact with different other structures, such as, for instance, tyrosine kinase receptors, and that leads to a non-genomic pathway via the MAP kinase pathway, again initiating different transcription factors. And finally, we also have a membrane-bound progesterone component that also leads to more rapid intracellular effects. In recent years, it also became clear that not only the classical pathway, the binding of steroids to a hormone-responsive element, and then activation of transcription is obviously important, there is also an epigenetic modification. So we know that progesterone may modify DNA methylation as well as histone acetylation. Both are important pathways of epigenetic modifications. And even more recently, it became clear that there is also a way after transcription to modify the translation into a protein. And we know that progesterone may induce different microRNAs that are small pieces of messenger RNA that interact with the messenger RNA and may lead to a partial stop or even a degradation of messenger RNA, and therefore a blockage of the translation into a protein. So these epigenetic modifications are especially relevant, for instance, during pregnancy. We know that progesterone, which is one of the most important substances to keep the uterus quiescent, when binding to a progesterone receptor, induces a specific transcription factor, which is called SEP1, zinc finger E-box binding homeobox, which is located at chromosome 10. And by inducing this transcription factor, there is an increase in microRNAs, the 200 family, and that in turn leads to a downregulation of contractile gene expression. And that is probably the mechanism why the uterus and the smooth muscle cells do not contract too much during pregnancy. But these microRNAs are also important concerning implantation. We know that messenger RNA stability is important in different aspects, such as, for instance, inflammatory, but also apoptosis, neoangiogenesis, and cell proliferation and differentiation throughout the whole cycle. So we have many different effects of progesterone. We have the reproductive effects, and we have effects on, for instance, the central nervous system. Recent studies have shown nice effects concerning neuroprotection, as well as an influence of progesterone on body temperature control. All gynecologists know that, and there is also an effect on respiration or pain perception. But today we focus on the reproductive effect, and progesterone is essential, especially concerning the secretory transformation of the endometrium, decidualization, implantation, as well as immune modulating effects that are involved with the implantation, and as shortly noticed, the contractility of the uterine muscle cells. So during a normal cycle, there is a lot of change in the endometrium. In the proliferative phase, there is an increase in estrogen production, leading to a strong impact on cellular proliferation and growth, as well as angiogenesis. Then there is the LH surge, and thereafter there is an increase in progesterone production, leading to a perfect so-called window of implantation, which is around 6 to 10 days after the initial LH surge. And progesterone within the secretory phase leads to a glandular secretion and the stromal decidualization, as well as immunologic effects. So in the proliferative phase, estradiol is essential for endometrial cell proliferation, and we have actually two different layers. We have the superficial epithelial cells and the underneath stromal cells, and there is a specific estrogen-dominated cytokine production in these two layers. But when progesterone production is increased, different regulators are induced. There are signaling molecules that appear like Indian hedgehog or HAND2, which is heart and neural crest derivative 2, and a specific cytokine pattern is present under the influence of progesterone that not only changes the epithelial cell differentiation as well as initiates stromal deciduation, but also is interacting and attracting immune cells from the circulation, such as macrophages or dendritic cells, and also the natural killer cells that are present within the uterus, leading to a receptive endometrium and successful implantation. If you watch the endometrium, the surface of the endometrium by electron microscopy, you see a huge difference between the proliferative phase, where a lot of mucin is produced from the superficial layer, whereas in the secretory phase, there is a complete change in picture. Here we see the so-called pinopods, which are protrusions of the epithelial layer of the endometrium and are the perfect place to interact with the trophoblast. So here you can see this pinopod that is interacting already with two different trophoblast cells. So the mouse model is usually a perfect model to investigate what actually happens during this optimal window of implantation. There is a lot of crosstalk, not only between the embryo and the endometrium, but also between the epithelial layer as well as the underneath stromal cells. And during a more estrogenized situation, there is a lot of estrogenic driven proliferation, as well as production of this mucin on the superficial area of the epithelium, which actually keeps microorganisms or also trophoblasts at a certain distance. And that is driven also by stromal factors such as fibroblast growth factor that induces especially the estrogen receptor. And then when progesterone is becoming more and more predominant, there is an increase, first of all, in progesterone receptors, and then via signaling molecules such as Indian hedgehogs, there is an increase in nuclear receptor factor 2, family 2. That in turn leads to a decrease in estrogen receptor within the epithelium, and also a decrease in fibroblastic growth factor, and that leads to a shift in a more progesterogenic situation, and further on via the bone morphometric protein as well as wingless and also HAND2, there is the initiation of decidualization right at the window of implantation when the embryo approaches the inner surface of the endometrium. So progesterone, and to a less extent also cyclic AMP, is transforming endometrial stromal cells by the initiation of different factors into decidual cells. And then these cells produce decidual-specific factors such as prolactin or insulin-like growth factor binding protein 1. And these decidualized cells then exert different activities that are important for the implanting embryo to facilitate implantation. First of all, there is a downregulation of radical oxygen species. There is also a loosening of the extracellular matrix by degradation of collagen. There is also an influence on natural killer cells. I come back to that in a minute. And there is an influence on neovascularization for the embryo to get access to the maternal circulation. Here, the trophoblastic invasion in a timely and gradually developing situation. First, pinopods appear. Then there is a lot of cytokine signaling, especially the LIF factor, the leukemia inhibitory factor, is upregulated massively during the optimal window of implantation. There are also receptors on the outer side of the cytotrophoblast for interaction. And then there is adherence of the trophoblast to the pinopods, a lot more of cytokine and chemokine production from both sides. And finally, there is the invasion of the trophoblast and the end of the window of implantation. So the timely and gradually network of progesterone-dependent factors is really necessary for successful implantation. And one of the most important trigger is the ratio between the estradiol production as well as the progesterone production, which is highest in that window of implantation, whereas the estrogen production is downregulated, at least for specific intervals, so the implantation may not be disturbed. There is other important impact of progesterone concerning the establishment of a successful pregnancy, and that is the impact on immunologic reactions. As you know, the fetus is actually a semi-allogenic tissue, and there should be a protective immune modulation of the mother, and that is also triggered by progesterone, because progesterone leads to an increase in so-called asymmetric antibodies that do not display cytotoxicity or phagocytosis. And there is also an induction of blocking antibodies to mask the fetal antigens. And then with progesterone, there is an induction of the so-called progesterone-induced blocking factor within the endometrium, and that leads to a shift from the more cellular to a humoral immunity, with the shift from Th1 to Th2, where different cytokines are produced within the endometrium. And finally, there is also an influence of this progesterone-induced blocking factor on the natural killer cells' activity to be downregulated to accept the semi-allogenic material. And that, again, can nicely be demonstrated in a mouse model that progesterone leads to decidualization, which improves trophoblastic invasion. Then we have, via epigenetic silencing mechanisms, a different pattern of cytokines, a shift from the Th1 to the Th2 state. We have the induction of progesterone-induced blocking factor that leads to immunosuppressive activities. For instance, galactine is increased, and we know that galactine leads to an arrest of the local dendritic cells. And finally, progesterone also modulates maternal immune tolerance by, for instance, increasing local recruitment and generation of T regulatory cells. And that finally is a prerequisite for a successful implantation. And finally, let me shortly address also progesterone throughout pregnancy. It's really impressive how high progesterone production is induced during pregnancy. We have really highest levels at the end of pregnancy in the serum of the mother. And interestingly, even though progesterone is massively leading to a downregulation of contractile properties of the smooth muscle cells, there is no decrease at the end of pregnancy in progesterone concentration in the maternal circulation. So progesterone, which is produced within the placenta in increasing amounts out of cholesterol, leads to uterine quiescence. And not only the parent compound itself, but also, for instance, the 5-beta-dehydroprogesterone is important also for down-regulating contractile activity. And of course, there are also other metabolites, such as, for instance, allopregnanolone, which interacts with the GABA receptor in the fetal brain that may be important. And there's the way for eliminating via the 20-alpha hydroxyprogesterone pathway. So progesterone increases during pregnancy, and the reason why it leads to a relaxation of the uterine smooth muscle cell is the induction, especially of this transcription factors SEP1 and 2, that leads to an induction of microRNAs, and that in turn leads to a downregulation of contractile associated proteins, of the master switch in inflammation, the nuclear factor kappa B, as well as prostaglandins. And there is also an effect on the membrane-bound progesterone receptor component that leads to a downregulation of intercellular calcium. So progesterone is actually a very strong calcium antagonist. And then at the end of pregnancy, due to not well understood impacts, probably an increase in oxygen tension and lung maturation in the fetus, there is a downregulation of this microRNA 200 family, and that in turn leads to an increase in contractile associated proteins, in nuclear factor kappa B. So the initiation of labor is sort of an inflammatory process as well as prostaglandins. So obviously at the end of pregnancy, there is sort of a functional progesterone withdrawal. So in conclusion, all the different areas I demonstrated may be potential therapeutic areas for exogenous progesterone. We know that different clinical studies gave us very nice results concerning recurrent pregnancy loss or repeated abortion. There should also be a support of the luteal phase, especially in ART cycles, because in ART cycles, usually estrogen production is much higher, and therefore the ratio between estrogen and naturally produced progesterone may be impaired. Also, in specific risk groups, there may be a benefit of progesterone application concerning preterm delivery, and of course, in the peri and postmenopausal period of women, where there is a natural decrease in progesterone, some of them may also benefit from progesterone therapeutic application. Thank you very much.
Thank you, Kristen, for a very interesting lecture. And thank you for finding the time to be with us today in such a stressful time in Europe. But there are a few questions here, and I'll try to summarize them very shortly if you can approach each of them. One of them, we know during the last few years that we can stimulate patients and get very proper eggs during the time of the luteal phase. So is there any direct progesterone effect on the oocyte during the stimulation?
During the stimulation, there's probably no effect on the oocyte. The effect of progesterone is mainly concerning the endometrium and the successful implantation. I don't think, or at least I don't know any effects directly on the oocyte.
As far as the implantation window, can you please estimate the duration that this implantation window is available for the embryo to be implanted?
Usually it's around 3 to 4 days, the optimum of the endometrial changes, and then it closes again. However, it's never definitely clear how long it lasts, especially in ART cycles, because you have sort of a disturbed ratio between the estrogen production as well as the addition of progesterone, and therefore this might impact also the endometrial receptive phase. So usually in a natural cycle, the window is around 3 to 4 days, but it may be longer or sometimes also shorter in ART cycles.
There is a paper published by de Ziegler years ago in which he characterized, or actually he mentioned, that there is a desynchronization between the glands and the stroma, which might impair the function of the endometrium and implantation of the embryo. And he thought that the rise of progesterone, whether it's fast or slow, might interfere with the development of the endometrium. Can you comment on this, if there is a difference between the development of the glands and the stroma based on the progesterone level and the way or the time it takes him to rise?
Yeah, it seems quite likely that sometimes there is a dysynchronization between different tissues within the endometrium, especially the glandular cells, which might also be impacted, especially through estrogens, because estrogens has a strong proliferative effect also on the glandular cells. And sometimes there is a desynchronization, especially when the ratio between estrogen and progesterone isn't optimal, and therefore this desynchronized glandular proliferation may impact also the successful implantation.
And the last question, please. There are several formulations in terms of progesterone using for the luteal phase and supporting early pregnancy. And this is the IM, the subcutaneous, the vaginal, and the oral progesterone. Can you please comment on this?
Usually I prefer natural progesterone for supplementation in ART cycles, or dydrogesterone, which is actually also the same molecule and just fits a little bit better into the progesterone receptor and therefore has an enhanced half-life. So progesterone or dydrogesterone to me is probably a better option than any other synthetic hormone that has to be also applied, for instance, intramuscularly, which is usually not a very nice option for the patients. So we are using either natural progesterone vaginally or orally in higher dosages, or dydrogesterone where you can lower the daily dose of intake.
Okay,
is that better?
thank you very much. Thank you again for being with us today. And
Thank you.
And I hope that the situation in Europe will come better quite soon. And thank you again.
Thank you, Seth, and greetings, Bob Gleicher.
Thank you.
He's in
Okay.
Austrian.
Okay.
Bye-bye. Thanks.
Bye. Thank you.
Thank you.